This is a production of Cornell University.
So the topic today, I want to share some of our recent advances in genomics-assisted breeding in the Octoploid cultivated strawberry. And my program is strawberry molecular genetics and genomics. So at the University of Florida, I have about 15 different research and education center, and one of the research and education center name is Gulf Coast Research and Education Center at nearby Tampa. That's where I am working now. So this is the overview for today's seminar. I want to introduce about the UF strawberry breeding and what about the strawberry genetic genomics research. Then, so I want to share some of the research about the trait gene discovery for disease resistance and fruit quality in the cultivated strawberry. So I don't want to give too much detail for how we discover QTL, but I can share some of the information how we use this QTL information go through all the way for candidate in search, and to use this information can be used for the new variety development. And also I want to share some of the information what our recent research about the mutational breeding approaches for variety improvement, such as somaclonal variation and CRISPR gene editing. So then I have a couple of slides what will be conclusion for all the slides I talked today, and then what will be the future perspectives in the future research. So University of Florida, IFAS, so Institute of Food and Agriculture Science. So the Gulf Coast Research and Education Center is, wanna click the laser pointer? Yes, so this is the map of Florida. And so we have several important horticulture crops such as blueberry nearby Gainesville, and the peach and grape, strawberry, and the tomato is the place, the Gulf Coast Research Center. We have these two breeding programs, and Citrus Center is working on this, the orange juice and citrus breeding program. And our strawberry breeding program has started in 1925. So 1925, there was not really breeding program, but mainly for plant pathology, the program, because the Florida, the environment was such a nice in winter time where grower grows the strawberry, so their disease pressure was really high. So that was the main reason the University of Florida, they created the pathology and breeding program. And so the 1952, the first strawberry variety named Florida 90 was released. And so far, 2021, so we have a total 18 the cultivar released. So this is the US map for overall strawberry production. And so because the California is the leading state, about 92% of strawberry production, but these are all these summer strawberry production. Everything in the winter time, the Florida is only the state that mainly produce or fresh the fruits in around the US. And also the other fresh fruits coming to Mexico. And in 2015, the strawberry farm gate value was about 291 million in Florida. And so this is the estimated Florida acreage, what the variety is currently growing by farmers. So the Brilliance is the major strawberry varieties that released by UF program. So about 50%, and Radiance about 10%. So Radiance was the actually major varieties about a couple years ago, but because of some of the the problems such as this is very sensitive to heat and fruit is not very good shape for early harvesting. So the brilliance is changed everything for radiance now. And the beauty about 5% and sensation, which is really the good size of fruit, and the sweet sensation is the highest bricks ever what we have from our breeding program. So about 10% is drill score. So about total 90% of the variety what growing in the Florida is released from the University of Florida strawberry breeding program. So what is our breeding for the breeding structure for new variety? This is pretty simple. So mainly it's a conventional breeding for disease resistance and fruit quality. And we recently add in this DNA-informed breeding technique in here. So this is the one picture. So I joined the UF strawberry breeding program 2015. So when I actually joined the first time, and this is we have DNA testing system we using one marker. So this one marker we called the FAD1 gene is the peach aroma gene. So when you look at these two different patterns of the DNA fragment, this is basically PCR, the dominant marker assay. All the way the bottom is the housekeeping gene control, and you see the DNA presence and absence based on the PCR amplification. And all this marker system we use as a marker assisted seedling selection. But our goal is really, we have so many different traits we want to combine, bring together, stacking into one main cultivar for disease resistance, fruit quality. But the previous, the gel-based system is obviously it's impossible we can do this. So currently, the program structure is from Dr. Vance Whitaker. He's a strawberry genetics and breeding, a strawberry breeder. And my program, strawberry molecular genetics and genomics. We have all different research section from trait to discovery, validation, and genomic prediction, and cultivar development to release. These three programs is led by Dr. Vance Whitaker, and then the marker assist breeding and genome assembly, genetic engineering. This all part I work on this. So this is the simple diagram for strawberry breeding cycle. And we have about 100 to 120 cross every year. And every seedlings, it's screened by DNA marker. And then after seedling selection, it goes to the advanced selection, clonal testing. But last 2 years, we add in the new method, genomic prediction, genomic selection. It can actually bypass these field trials and go directly to year 3 cross. And also, this marker can be used for parental screening. So this is typically from year 1 to year 4, and year 5, we have the one total the breeding cycle. So using this cycle, we normally have around 5 to 6 years to have the strawberry cultivar. And this is a very simple, the kind of direction for how we use a strawberry DNA test. And so we have all the seedlings planted in the pea pellet, and this is all the structure is exactly like PCR 96 well plate. The tiny leaf disc, we can do direct rapid DNA extraction. This is NaOH based, and then all this the dirty DNA, but we have some other way we can do direct PCR and genotyping using all this in-house high resolution melting system. And this is typically what we have the plates at the end of this marker seedling selection. So what about the traits we work on the new cultivar development? So these are typical the disease problem. Angular leaf spot. This is only the bacterial disease in our the in Florida situation. And Phytophthora crown rot, collector trichome crown rot, powdery mildew, Anthracnose fruit rot, charcoal rot, botrytis, fusarium wilt. And recently we got another big problem, the leaf spot and fruit rot. This is a new disease. So Neopestaloptiopsis. So this is really spreading everywhere this year. So this is a new pathogen. And so which pathogen is more important? I cannot really tell. Everything is come up together because of the so favorable weather to pathogens. So we have to work all these disease problem. Basically, we have one PhD student, master student, or postdoc. They have one trace, everyone. And also we work on the fruit quality for their neutrality. This is getting more and more important because of early harvest and planting in hot condition, early harvesting. And also gamma-D-galactone is important compounds for fruity aroma. And also fruit color is one of the important traits. And so recently we study more and more on the flavor and fruit quality. So methyl anthranilate, that is the grape-like flavor is something very unique and interesting, the flavor. And mesifura is a sweet and sherry-like flavor is very important. Also linalool, that's like a floral flavor, and many of the consumer panel really like all these compounds. So I divide about 4 different sections, and then so want to introduce how genomic information we use for breeding program. So the first, the title is about how DNA technology and genomic information can be utilized to improve strawberry varieties. So I want to share a little bit the background about strawberry genetics. So cultivated strawberry, as everyone knows, is Octoploid strawberry. So it has a FaRCg, a family Rosaceae. So the cultivated strawberry is the scientific name is Fragaria and Ananasa, and crossed by 2 Octoploid wide species, Chiloensis and Virginiana. So there are total 7 chromosomes and 4 different subgenomes. So total chromosome number is 56. So this is origin and evolution of strawberry published by Dr. Patrick Egger at Michigan State University, 2019. And the first tetraploid strawberry was happening here, the Asia, China, and Japan, Korea area. So Fragaria nipponica diploid and Fragaria inume diploid, they naturally crossed, and tetraploid crossing by viridis and become hexaploid and moschata. And this moschata all the way come into the Northern America area and met Vesca. So this is the first time happened the octoploid, the wild species Virginiana in Northern Virginia, the area. And these Chiloensis and Virginiana went to the Europe by European gardener. They crossed by gardening strawberry. That the first name become Ananasa. So the fruit is more likely commercially big, and the quality is more like a fruit. So this is currently what we cultivate the mostly in US and also other European countries. So the Vesca derived genome was the first completed 2011, 10 years ago. So this is really breakthrough for strawberry genomic research. But still, this is only one subgenome for Octoploid strawberry. In 2019, the Fragaria ananasa is the cultivar from UC Davis line, the Camarosa, was the first time complete genome assembly, and the gene number is over 120,000 genes. That's a lot of genes. So these 2 information actually it's very important to use the breeding program. So we can make a SNP array chip, can do genotyping, and can do QTL discovery, and further move on many other things. So the 90K axiom SNP array developed from VASCA, and the other 50K FANA SNP array developed from NASA's Octoploid. So I can share some information what these two array results can be different from next slides. So this is one example. We use the 35K, 90K, the array developed from VASCA, and identify QTL for collector trichome crown rod resistance. When you look at this background picture, this is the field trials. There's some other plants are really healthy, but others are really dying, just collapsed because of this collector trichome gloeosporids. That's the pathogen. And what we did, the 3-year field trials, we crossed in different populations testing. We found one major locus at chromosome 6. It controls the resistance, the crown rod resistance. And this is 35K already, the data. So that's why we cannot tell the subgenome, but we can tell chromosome because of based on the VASCA, the chromosome marker data. So we have to identify what subgenome is for this the location. So we use the Flex QTL mapping. This is software. So using this software, we can make all linkage map for each subgenomes and find what SNP markers located the target unique subgenomes. Then can tell 6B is the one that where CG1 is located. And this is the one of the disadvantages to use the VASCA SNP marker. We cannot, it's difficult to identify subgenome location. And also when we identify this location of QTL, the QTL information is too much broad. We cannot use the marker for this, for the seedling selection. So that time we had to use RNA-seq data and find the candidate genes in this CG1 region, which gene not involved, then we can actually narrow down at the CG1 region, and we found these are the 3 candidate genes are highly expressed on this resistant line, the Eliana. And then we can actually using the short read of RNA-seq and using Camaros reference genome, identify exact the unique subgenome, then physical location of CG1. So this 1.2 megabase region is the original primary mapping of CG1. This is too big to develop DNA marker. So typically in my experience, less than 500 kb is the target region, the DNA marker we develop for DNA marker that works very well. It's about 90% accuracy marker prediction. So in this, we narrow down 500 kb region, identify these all the candidate genes. And this marker we developed for the target candidate gene receptor like kinase 1A. There's a unique in there between 2 resistant Eliana and Winterdon, 2 other susceptible accessions, sensation and Camarosa. You can develop marker, the high resolution melting marker. You can really predict resistant and susceptible the seedlings. Then, so we look at the whole this population, what is the marker accuracy and the validation of selection efficiency. And this marker really highly co-segregate with the phenotype resistant and susceptible. So still, when we think about strawberry genome, it's complex and it's highly heterogeneous, but it's somehow we are very lucky. Many of important traits for disease resistance or fruit quality is controlled by one or few genes actually. So like Anthracnose flora, we found one major locus control for the resistance, and also Phytophthora crown rot and bacterial angular leaf spot. Everything is one locus. And for charcoal rot, we found 3 loci is necessary. So each loci actually about 30%, 20 to 30% phenotypic variation. So we have to combine all 3 together. Powdery mildew is really multigenic. We cannot do the marker-assist selection, but we use a genomic selection. So if possible, I want to share all the information, but because of time limit, I cannot do. But genomic selection, it works very well. It's a very interesting result also we have. So, but I can share some of the information, powdery mildew, how this is the important, the mildew locus is associated with the powdery mildew resistance. I can share a few slides later. And botrytis gray mold, this is important disease problem, but we don't have any genetic resistance. It's not just our program. I don't think any other strawberry program has a genetic resistance source for this botrytis gray mold. And other this, the FAD1, the fruity aroma flavor also controlled by one single gene called FAD1 gene. And also we recently found the red coloration of fruit, it was white fruit, is controlled by one single gene MIP10. And also there are some of the important flavor, linalool, mesifura, methyl, anthranilate. This is our controlled by 2 or 3 different loci. So this is one of example powder mildew, the resistance, how we use the genomic data we use for the improvement. And so the powder mildew is one of the important disease in Florida because of hot and humid weather, especially in the December and March. And so if we don't control this pathogen, it can actually cause up to 60% yield loss. So based on the Kamarosa genome's the reference sequence, we did genome-wide characterization of MLOs in Octoploid strawberry. So the A in the top, these are the VASCA MLO genes, about 20. And at the bottom, we actually look at the all MLO genes in each subgenome level and the 7 chromosomes. So there are a total of 68 MLOs in Ananasa. And these all phylogeny trees don't have to really look very detailed, but what I want to show when we look at each grouping here, so the VASCA or the diploid, the MLOs are matching very well with the Ananasa, the octoploid MLO in same chromosome. And also we see the clear pattern of the VASCA. VASCA is a dominant subgenome compared to the other 3 subgenomes. So we see the VASCA MLO are always present with the Ananasa. So jumping on that, and what will be, so there are many, the reports are there, studies are there, the MLO, this gene is a recessive gene, is important for powdery mildew resistance. And also we wanted to look, there are so many MLOs, so what are they doing for powdery mildew resistance? So we look at these 2 resistant genotype, the accessions, and 2 of the susceptible. We compare the transcriptome data analysis. So these are all differentially expressed 68 MLO genes between 0 and 24 hours per infection. We look at which gene is up and down regulation. We found the MLO 10 and 20 was consistently the up regulation in susceptible, 2 susceptible. And the 2 resistant accession doesn't have much expression in MLO 10 and 20. And this is the QRT-PCR. We found the same pattern especially for 24 hours after infection. For MLO 17, we see that there's a highly up regulation in susceptible accession, but somehow we didn't see that this much data, RNA-seq data. And also this MLO10 and 1720 was published in other crops for the involvement of the powdery mildew resistance. So we look at the RNAi transient assay for these two genes, and the MLO10 and 20. So this picture show MLO10 and 20 is successfully downregulated in our RNAi transient assay. So what we did is just whole plant to the vacuum infiltration, and 5 days later, we did a powder mildew, the inoculation. So when we look at the disease progression, it's empty vector, and this is MLO10 and 20, MLO10 and 20, both that we knock down together. So in the disease severity index, we look at the empty vector is always high. As you see the probe discoloration on the surface of a leaf, but this MLO10 and 20 and 10, 20 together, this is even transient assay, we see the disease progression is really down. So this is the kind of the indication the MLO10 and 20 is important for powder mildew resistance. And now we are moving on the CRISPR knockout that the experiment. So the second section is, so how could genomic information be utilized for to enhance fruit quality and flavor the Octoploid cultivated strawberry? So this is the white strawberry we just released last year. And so this white strawberry germplasm coming from Japanese, the variety, we cross our line, and then this is kind of pedigree. We made the population, we look at all the phenotypes data. And so this is really one of the power to use the 50K SNP array. And we can tell exactly subgenome. We don't need to do any more the QTL, the flex QTL to identify subgenome. Also, this SNP, what we developed, this SNP array, we developed at least 3 SNPs every gene in ananasa. So and if there's such the high level of SNP, we can use directly for candidate gene identification. So we did MSeq for this MIP10. We found very strong SNPs in these MIP10 candidate genes. We found there's the A base pair insertion at the white. And based on this marker, we can exactly tell white and red fruit. And this is the collaboration with the Spanish group, and we published the plant cell paper. And so in their population, they identified in transposon in promoter region, and they actually telling red and white exactly segregating. And also the bottom picture, what is showing that anything what insertion and mutation in the exon 3, it makes it completely white. So there's one actually question for me here. I understand MIP10 mutation make white fruit, but what about the finger fruit? We have no idea. We have the segregating population from different pedigree. It always shows as a really nice pink, as rosette pink, but we don't know actually what's happening here. So this is the one of study we are continuing now. So one of the important fruity aroma. So at the reference genome does not have this the FAD1 gene and gamma D-galactone gene. So this is a kind of problem. If you have a reference genome does not have a genomic information, you cannot really work on that, and you can really characterize what this genomic region is doing, and it's hard to develop DNA marker. And also one of the other important question is, even though FAD1 gene is present all these assertions, but there's great amount of the gamma D-galactone, the contents, but this is really absent because of the FAD1 gene is not there. So that was our question, what's the happening? But this is kind of structure to characterize and cloning FAD1 genomic region in Octoploid cultivated strawberry. So we did the GWAS study, typically what we do for some of the traits. And since the reference genome does not have this genomic region, so we did a hybridization-based target enrichment sequencing. It's a probe capture sequencing. Also, we did the back library sequencing. This is based on PCR, the screening method. So And so based on that kind of research approaches, we identified this Kamarosa region, FAD1 gene is missing using the back clones. We did all the Illumina sequence short reads combined together between producer and non-producer. We found the large gaps. All these things is non-specific short reads from other subgenome or other homologous regions. So we don't worry about this one. And we identify exactly 8.2 KB region is missing at non-producer. So based on that information, we develop codominant marker. So it tells exactly heterozygous and homozygous non-producer, homozygous producer. And we also found the gene dosage effect. So when 2 copies, homozygous always more, and heterozygous almost about half. So based on our all volatile contents, GCMS data is strongly suggests that. So what about this gamma D, the accumulation, the variation actually is caused by gene dosage effect. And the other approach, what we do, so this is really kind of a little more the complicated process. This is a haplotype-phased genome assembly. So the like gamma D or FAD1 gene was, we use back library, we use different approach because the haplotype-phased genome assembly is technically not easy and not simple. There's a lot of things we have to make bioinformatics scripts, everything we have to test. And also because of heterogeneous and subgenome issue, we have to make our own the pipeline. So it took about 1 year. We complete this bioinformatic process, and now we can actually assemble all any Strawberi Octoploid Strawberi session in 1 week when we have all the data sequence. So this is kind of an example how we do haplotype-phased genome assembly. Basically, we use for this method for the fruit flavor because of fruit flavor is more complicated, and also the pathway more other gene interaction. We want to look at the whole genome level and go back to our target, the genes or loci controlling the flavor. So we do the PacBio equal to hi-fi CCS reads. This is about 99% sequence the accuracy. And also we have to also use Illumina the read for all this, the phasing assembly and error correction. And while we use the hi-fi ASM, this is the one method we use. It works pretty well in scaffolding. Then using, after we do scaffolding, haplotype phasing, all this genome quality evaluation we test, and all the misassembly correction. So this is the very simple way we have for haplotype phased genome assembly, we must know the immediate parent of the genotype or accession you wanna do assembly. So this is how we do hi-fi reads, 2 smart cell, about 40x coverage, 1 accession, and these 2 parents we do only the Illumina short reads, 30x. Then we can do the exact haplotype assembly. So this is the result one, the beauty haplotype. Or the haplotype, it really good linearization. That means the quality of a single chromosome haplotype is really good. And also this is the one haplotype, the other haplotype, there's nothing overlap each other. That means the unique haplotype 1 and haplotype 2 for every chromosome level, that the quality is really high. Then we compare to Kamarosa versus the haplotype, the assembly data. When we look at the M50 value, it's about 12 megabase, and each haplotype in Kamarosa is very short. So, and also the Vosco value, this is the telling the quality of assembly is almost 98% versus 96%. So our assembly data is really good quality. And so that's the methyl Anthranilate. Also we did, we did also the haplotype genome assembly for Mesifura, and also it tells the similar, the higher result, about 98% haplotype 1 and 2. the quality. So I have a few more slides. What alternative breeding approach we can really improve strawberry variety other than just genome data and sequencing information. So we use somaclonal variation, CRISPR gene editing. So this somaclonal variation breeding is typical. We has a tissue culture process. So we don't do any the mutagen treatment, just the hormone level. We adjust different hormones. And this is runner cut. It go all the way process. We just do shooting regeneration, and we collect every somaclones. Then so this is one of the example what we test in the field. So this sweet sensation FL127, it's very good. Fruit, sweet, and the growers love, especially local market people, they love it. This very. But one of problem here is bush shape, and everything is hiding in here. So fruit is difficult to harvest. The harvesting time is slower than the cultivar, and also it's difficult to manage. But it has a more higher nutrient, the nitrogen and something else. And these are all the soma clones we identified has a smaller plant size, compact size, and has a nice fruit, the appearance. So this is something the kind of primary result what we have. And the other interesting is Phytophthora clonal resistance. We just screen every the soma clones. We identify some of the soma clones is not dying, and this is a positive control. And this is something we are interested in, and we continue to screen for these resistant soma clones. And we have 2-year field data for these soma clones, and they're consistently resistant. So that's interesting. So another one is heat stress tolerance. This is a recent study we moved on. And this Florida Beauty is early planting for early harvest. And we did all these soma clones did heat stress in test tube. And you look at here, one plant, probably some of the plants back there, but it's not dying, it's survived. This is really 7 days and over 40 degree at really strong heat condition. And when we look at the control of the 2 different resistant group, we always see the root elongation, the root is the actively grow. And this is a greenhouse test. We did soma clones, we propagate, and greenhouse test will really see in the adult plant the different level of susceptible to intermediate and highly resistant for heat. So that's interesting that we are now moving on the field experiment the coming season. What about the CRISPR gene editing? What we are using this one? So this PDS gene knockout, this data, this is kind of we test our CRISPR gene editing system and vector construction in Octoploid strawberry, and we found it works pretty well. And so this is now we are actually using this MIP10 knockout and change this the fruit color, and also we want to show exactly how we can target unique subgenomes, this MIP10. That's always people question about how you can knock out single gene. It's such a specific the subgenome. So that's what we are testing in this whole experiment, and we want to publish out the data. And we have now these some of the plant in the rooting media. And so as I mentioned earlier, the botrytis, it does not have any genetic resistance source. So this CRISPR could be a really good method to do that. And so we identified the one candidate genes, wacky gene from public database search, and we screen all the Arabidopsis mutant using the strawberry botrytis pathology. We identified the one wacky gene, and actually it's 2 copies in the chromosome. We did the RNAi. When you look at the wacky 25, wacky 45 is significantly delayed compared to empty. And somehow when we knock out both, silencing both, it's actually more disease than the single silencing. So it's interesting. And also this frequency always show 4 days, 5 days, 6 days after infection, always a lower infection rate. And this disease area actually looks like 45 is a lot better. So this is kind of CRISPR approach. We're gonna move on. So summary for this whole, a lot of different things in this talk today. And so DNA marker and testing is the kind of important the program, because everything what we use sequencing and the trait discovery, this is, we want to develop DNA marker and use that for integration of that new traits. This is resistant and prog. So typically every year we do 83,000 seedlings we screen in 5 weeks, and total number about 12,500. This is what we sent for Californian summer nursery for coming the breeding season. So right now we use about 9 different markers for day neutrality and gamma-D-galactone flavor and white fruit, other 6 disease resistance traits, and the phase genome assembly. So eventually, actually, we're gonna make a high quality of chromosome scale reference for Florida brilliance. So this is gonna be our reference genome. And also we're gonna do re-annotation of Octoploid Strawberi using iso-seq transcriptome data. And also you will also add into the current draft Octoploid genome. We put the high quality of another reference genome. Also, we're gonna make a 4 haplotype reserved genome. So this is more like fan genome, the structure for flavor and disease resistance. So we're gonna continue for Strawberi, the Somaclonal variation and CRISPR gene editing for Phytophthora crown rot, heat tolerance, and also herbicide resistance is one of the new traits. We're gonna work on the Somaclonal variation. And also these are all other traits for CRISPR gene editing. So I have some interesting questions for future perspectives. And so we have still keep coming the new disease and new traits coming in. So we need to continue for QTI discovery and gene cloning. And also we recently started our 6 SNP working with LGC. So this is the more on for genomic selection we use for that, because of the price is currently too high. We cannot screen all our seedlings. And also Strawberi pangenomics and systems genetics, it can be eventually very helpful flavor the improvement studies. And so DNA marker and test, we're gonna continue to develop for multiplex and also automate genotyping. And also CRISPR, somaclonal variation, or the mutational breeding approach can be used. This is a new disease problem because it's getting more and more important. And also we are also working on the transgene-free CRISPR gene editing method. This is coming probably next few years later. So thank you for listening for all my talk today. And then so this is a really great team for the Dr. Vance Whitaker group and my group. We work all together. And so this is how we develop new strawberry, the variety for strawberry industry and the growth. And I have also acknowledged to Florida Strawberry Growth Association and Florida Foundation Seeds, and also the USDA, the Rose Breed, and also Florida State. All this fund was done for this research. Thank you for listening. And so I want to have some other questions you have.
I think we have about 5 minutes for questions. I'll ask one of Dr. Lee. I noted that when you were indicating various qualities that you are breeding for, that one of the scent qualities was linalool. And I was wondering if you're familiar with work that's been done on linalool that shows that it has an active, relaxing quality on the brain.
No, I never heard that. Thank you.
Work that was done at Kagoshima University in Japan specifically on lavender, but it is interesting to learn that strawberry also contains linalool.
Yes, so right. So when I taste the certain varieties from field which has a more linalool contents, I feel more relaxed. Yeah, that's I agree there. But yeah, but
Well, now you know why.
yeah, I feel like pleasant. Yes,
Right.
But the other compounds, methyl anthranilate, is a grape flavor. That one is really, some people, other group like it, other group does not like it. Yeah.
Well, let's see, we have a chat comment from Bruce Reich who says that grapes, especially the muscat types, have lots of linalool. So another way of relaxing.
You know, that's correct. Actually, we found one QTL in chromosome 7 overlap. The candidate gene overlap the methyl anthranilate and linalool. Yes, that's the certain genotype we saw that. Yes.
Thank you.
Hi, Dr. Lee. May I ask a question?
Yes, sure.
Hi, Dr. Lee. Great job. I enjoyed your seminar very much.
Thank you.
I have a very quick question regarding your SV, actually, soma variation. Is this actually, it seems to me, actually, the frequency is way too high. Is it because it's specific to this strawberry, or it's a general, well, generally it shouldn't have such a high frequency?
Yeah, we actually, I surprised too. It looks like strawberry. Yeah, Octoploid strawberry. Because Octoploid strawberry is, we cannot do selfing because of severe inbreed depression. And also the strawberry domestication is about less than 200 years. And I think genetically it's still unstable. So that's what we see, that somatic variation ratio is quite higher than the other crops. Okay. And also we do also asexual the mutation. We got nursery, the industry, they don't propagate over 4 years because of asexual mutation. So we send the mother plant every 4 years. That means it's naturally the strawberry produce the higher the somatic mutation.
I see. Okay, but have you sequenced that, for example, that Hedgehog, well, heat-resistant actually genome?
We have not done yet, but we are planning to do for heat stress.
Okay, great. Thank you. Great job.
Dr. Lee, there is a question on the chat from Shantanu.
Yes, the question is, you mentioned that quite a few of genes are controlled by a single genes. Is that the case for firmness as well in strawberry, more specifically post-harvest maintenance of fruit firmness? Fruit firmness is not. It's not by a single gene. We found at least 3 loci are involved for fruit firmness. And also fruit firmness is 2 different. One is firmness outside the layer surface, and also firmness more internal firmness. It's a complicated one.
I think we have time for one more question if anyone has one.
I really loved your talk. Thank you so much for sharing this research with us. I can't believe you created the white strawberry. That's just so neat.
Right, that tastes very good actually. It's peachy taste and pear taste.
Wow. Oh my gosh, I have to try one. But my question for you is this. I think that it's very hard to conserve the aroma of a strawberry for it to last long enough to test in the lab. I was just wondering how you did that.
You mean how you keep the flavor for research?
Yes.
Data analysis. So we freeze right away. So when we harvest in the fruit, yes, we bring the liquid nitrogen and freeze right away at that keep in -80. And so, yeah, And then we don't want to wait too much for GCMS, the volatile data analysis.
Thank you so much for telling me.
And Dr. Lee, I think we have one final question from Courtney Weber in the chat.
So are you worried about concentrating your germplasm with a single gene disease resistance, increasing the incidence of disease breaking? Yes, yes. So this is a single locus, but I don't know it's a really single gene or not. I have some of the interesting results to share, but don't have time. And I don't think it's a really single gene. It's a one major locus. There are at least the Phytophthora, crown. I found 3 genes are involved together. And also there are 2 different haplotypes from 2 different origins coming together and get together. And even chocolate, we found 2 different subgenomes in one same chromosome. They actually show resistance. Actually, that means one subgenome from Vesica, the other subgenome from Niponica. So that's the 2 different diploid originate coming together. So, but there's not much study done for pathogen population. And especially in Florida, we don't have the broad population structure for pathogen because summer is so hot and humid, the pathogen is not really surviving. But maybe North Carolina and New York, that area, probably it's a more things. Yes, I think that's something important to think about, the breakdown, the gene.
So it sounds like that may require an additional dialogue between Dr. Lee and Dr. Weber. But thank you so much, Dr. Lee, for your presentation today. And thank you, Seonghee Ban, for introducing us to Dr. Lee. Have a very good week, everyone, and we will see you again next Monday.
Thank you, everyone.
This has been a production of Cornell University, on the web at cornell.edu.